students, and today we are going to start with the new topic, the biomolecules. So what are biomolecules and why do we have to study the particular topic? So in the last unit we have studied something about the cells. So what are cells, why we are calling this a cell, and what is the composition of the cells? We have studied something about the cells. And in this particular unit we are going to study about the chemistry of the cells. And cells are made up of, or the composition of cells. And why do we have to study the particular chemical composition, or how can we study the particular chemical composition of the particular cells? And how can we study? That is what called chemical analysis. Suppose you are taking a living sample, for example some tissue. What is tissue? Tissues are group of cells. Okay, any tissue. It may be a leaf, or it may be a meat sample, or it may be a fruit sample. Any tissue you are going to take and you are going to analyze by means of chemistry. You are going to do the chemical analysis and you are going to list out the chemicals present in the, you are going to list out the chemicals present in the particular tissue sample. Okay, and you will get the list of chemicals. In other term, you are going to get the list of elements present in the particular tissue sample. Okay, for example, you will get the list like this: carbon, hydrogen, oxygen, nitrogen, sulfur, magnesium, calcium. Okay, like that you will get a list of chemicals or list of elements present in the particular tissue sample. Suppose you are going to take a non-living matter or non-living sample. For example, you are going to take a earth crust. You are going to take a earth crust, some sample from soil, and you are going to do the same analysis, same chemical analysis, and you are going to list the chemicals or elements present in the particular non-living matter. And here also, for your surprise, you will get the same list of elements: carbon, hydrogen, oxygen, nitrogen, sulfur, magnesium. Almost you will get the similar or identical list of elements or chemicals you will get from the both living and non-living samples. Then how can we differentiate these living from the non-living matters? Okay, so according to the chemical analysis you will get though you are getting the same list of chemicals, the composition, the abundance of chemicals will be vary from the non-living matters. For example, in case of living cells, in case of living tissues, the common elements or the abundant of elements are the carbon, hydrogen, oxygen and nitrogen compared to the non-living matter. Okay, so over here these elements will together will make a compounds called organic compounds. So crudely we can say any carbon containing compound as organic compounds, but that is not 100% correct. So the point you have to remember over here, the organic compounds are comparatively abundant in the living matter than the non-living matter. So as I told you, in any living sample, in any living tissue, you can take the sample and you are doing the chemical analysis and you will get the two kinds of samples. You will get the two kinds of results. It may be organic elements or inorganic elements. So before we are proceeding into the list of organic and inorganic elements, how can we perform the particular chemical analysis for the particular tissue sample? For example, as I told you, you are going to take a tissue sample. Okay, you are going to take a tissue, any tissue. Okay, then you are adding something, some substance, some acid. For example, you are going to add some acid, trichloroacetic acid. Okay, you are going to add trichloroacetic acid, short form TCA. Okay, and you are going to grind your sample by means of mortar and pestle. Okay, it's a grinding stone. You are going to grind your living tissue or sample along with the acid and you will get a paste. Then what you are going to do, you are going to filter the paste with the help of any cellulose material or some filter or some. Okay, you are going to filter it and you will get two portions. Okay, one is filtrate plus the remaining things, the sediments. So filtrate means they have dissolved in acid, acid soluble and acid insoluble. You got the point? You have taken the sample, living tissue sample, and you have grinded with in the presence of acid, trichloroacetic acid, with the help of mortar and pestle, grinding stone, and you have grind it, and you will get the paste, and you are squeezing the particular paste, and you will get the juice out of the particular paste. That juice is called filtrate, that is acid soluble fractions of the particular living tissue sample, and insoluble fraction also you will get. And now we are going to analyze the components or the chemicals present in the particular acid soluble and insoluble fractions. Suppose, what may be the composition of this acid soluble fraction? As I told you, anything that can be dissolved in the particular acid will be in the acid soluble fraction. Okay. And what may be the composition of this insoluble fraction? Anything that cannot be dissolved in the particular acid, trichloroacetic acid. So normally in the insoluble fraction you will get the macromolecules such as proteins, nucleic acids, lipids, and carbohydrates. Okay. Almost all the macromolecules you will be see in the particular insoluble fractions. Almost all the smaller molecules and acid soluble molecules will be in the particular filtrate. Okay. And as I told you, now we will see the rough composition of a cell. As I told you, it is made up of both inorganic and organic. Okay. So what may be the percentage of inorganic molecule in a cell, or what may be the percentage of organic molecule in a cell? You may think the percentage of organic molecules are more in the cell, but that thing is a wrong thing. Okay, the point is the percentage of water is approximately 70 to maximum of 90%. That means water is an inorganic compound. Okay, just remember that there are no carbons in the water molecule. So water is an inorganic molecule. So water composition is the highest composition in the any cell, any living cell. Okay, so minimum 70% and maximum 90% of the composition of any cell is made up of water. So apart from water, as I told you, the next major compound present in any cell are proteins. It will be around 10 to 15%, and the remaining things will make up the remaining composition of the cell. For any cell, what do you mean by dry weight and wet weight? Wet weight means you're gonna say the weight of a particular cell or weight of the particular tissue with the water. Okay, that is what called wet weight. What do you mean by dry weight? You're gonna burn up all the water and you're gonna say the remaining weight of the tissue, that is called dry weight. So in a dry sample or in a dry weight, the proteins are the major molecules present in any cell. In a wet weight compound or in a wet weight cell, the water molecules are the major molecules present in any cell. So this is how we can study we can do the chemical analysis of the particular cell. And as I told you, you will get the organic fractions from the particular acid analysis. How can we get the inorganic compounds from the particular same tissue? Again, you will take the tissue. Okay. So what you have to do? You have to prepare the dry sample. So how will you prepare the dry sample? Suppose you have taken a leaf sample, you have to completely dry it so that the water from the particular leaf sample will be evaporate from the particular sample. Suppose you have taken a tissue, a living tissue like a meat sample or any other organ sample, you have to burn it. So what will happen while you are burning the particular living tissue? It will burn out. What do you mean by burn? All the carbon containing molecules will get oxidized into carbon dioxide. That means all the carbon containing compounds are burned. Okay. So what will remain? The remaining thing, ash. So what is the composition of the particular ash? The ash contains all the major inorganic molecules of the particular cell. Okay, like calcium, magnesium. Okay, so like all the major inorganic compounds will be present in the particular ash. Okay, so So in this way we can analyze the inorganic chemical composition of the particular cell. Okay, so till now what and all we have seen? We have seen what are the biomolecules. Okay, so what are biomolecules? In the biomolecules we are going to study the chemical composition of any living cell, and we are going to perform the chemical analysis to analyze the composition of the cell. We can do the acid soluble test to perform get the organic compound list, or we can go for the burning experiment to get the ash that contains all the inorganic fraction of the particular cell. Okay, and now we are going to learn two other terms related with the biomolecules, that is called the metabolites. So in the metabolites there are two categories, primary and secondary metabolites. So as I told you, the proteins, nucleic acids, lipids, carbohydrates, in other words the amino acids, the nucleotides and the fatty acids and simple carbons are the major components of any cell. Okay, so without these molecules a cell cannot function as a cell. Okay, so we are calling these molecules as the basic molecules of the particular cell, and these molecules together is called the primary metabolites of any cell. Apart from primary metabolites, most of the cells, especially the fungi and plant cells, they are going to produce some other compounds. They are going to secrete some other compounds. For example, some essential acids, some essential oils, some pigments. For example, we are going to use some perfumes or scents and rubber. So these are all, and fungi and bacteria, they are going to produce some antibiotics and some toxins, and these are all called secondary metabolites. Okay, so they are not part of the essential thing of the particular living cell, but they particular plants and fungi and bacteria, they need to survive. For survival they need these molecules, these rubber, pigments, essential oils, antibiotics, toxins, and we are calling these compounds as secondary metabolites. Okay, without these compounds that particular plant and bacteria can survive, but they need these additional molecules other than these primary metabolites, so that we are calling these compounds as secondary metabolites. So normally these molecules are involved in the human welfare. That means we are going to use these compounds for the human need. Okay, so the point you have to remember over here, you have to know the terms primary and secondary metabolites. So what are the terms we have learned till now? The chemistry, begin the biomolecules, the organic compounds and inorganic compounds, the percentage of organic compound and inorganic compounds, the primary metabolite and secondary metabolites. And before we are entering into each and every type of biomolecule, I will tell you two other terms: biomolecules and macromolecules and micromolecules. Micromolecules and macromolecules. So I told you in the acid analysis, in the acid chemical analysis, you will get the fractions, you will get the filtrate and insoluble fractions. Okay, normally in the soluble fraction, the filtrate, you will have the compounds with the molecular weight, below 1000 Dalton. Okay, we all know the Dalton is a unit of the particular, the molecular compound. Okay, so anything below 1000 Dalton will be called as micromolecules. So anything below 1000 Dalton will be called as micromolecules. Anything above 1000 Dalton will be called as macromolecules. So over here, proteins, nucleic acids, lipids and carbohydrates are bio-macromolecules. Why? Because their molecular weight normally larger than the 1000 Dalton weight. And among the bio-macromolecules, almost all the compounds are polymeric substances, polymers. And we are going to see the details of polymers later in the particular unit. So polymers are made up of monomeric units. Okay, the monomers are linked with each other and they will form a large unit of the polymer. Okay, so almost all the compounds of the bio-macromolecules are polymers except the lipids. Okay, so we cannot say the lipids as polymeric substances because we will explain. Okay, So the structure of lipid is completely differs from the remaining structure. Lipids are not a polymeric substances, but we will call this lipid as a particular biomacromolecule. Why we are calling this as biomacromolecule? Because their size is comparatively larger than this particular below 1000 Dalton molecule. Okay. So and these are the terms you have to remember with the introduction of the particular biomolecules. Biomolecules and the chemistry behind the biomolecules, inorganic and organic chemical elements, and the chemical analysis, the acid soluble analysis and the ash, the Barnett analysis. Then we have studied the primary and the secondary metabolites, then biomolecules, biomicromolecules and biomacromolecules. And now we are going to see the each and every type of biomolecules one by one with their structure and their function role in the particular cell.